There's a specific kind of exhaustion that doesn't come from working too hard. It doesn't come from not sleeping enough or skipping the gym. It's deeper than that. A flatness that shows up in your forehead around 2 in the afternoon. A reluctance to start things. A sense that you're moving through the day slightly behind yourself. Most people assume this is just aging, a natural dimming, something to manage rather than reverse. But the biology tells a completely different story. What we now understand, and this has accelerated dramatically in the last decade of research, is that the energy decline most adults experience in their 30s, 40s, and 50s is not primarily a function of age. It's a function of signals. Biological signals that have drifted out of calibration. And signals, unlike structural aging, can be reset. In the next several minutes, we're going to move through the actual mechanisms driving this energy loss. Not surface-level fatigue, but the cellular and neurological processes underneath it. We'll cover why your mitochondria are likely underperforming and what drives that. How light exposure at specific times can literally reprogram your circadian hormonal cascade. Why the timing of movement matters as much as movement itself. And how 2 underappreciated protocols, one involving temperature and one involving fasting, can activate systems your body hasn't used efficiently in years. These are not supplements. These are not hacks. These are the inputs your biology was designed to receive and has probably been starved of. Let's start where the story actually begins, inside your cells. Your mitochondria, and you have somewhere between 1,000 and 2,500 of them in every cell of your heart and brain, are not just energy factories. That's the introductory biology framing, and it undersells them dramatically. They are signaling hubs. They constantly assess the environment. Is there light? Is there food scarcity? Is movement happening? Is there a thermal challenge? Based on those inputs, they either upregulate their output, producing more ATP, the molecule your body uses as fuel, or they downregulate, throttle output, and begin a process researchers call mitochondrial drift. Fewer mitochondria, less efficient electron transport chains. More oxidative stress as a byproduct. Dr. David Sinclair at Harvard Medical School has described this process in detail in both his published research and his 2019 book. The core finding is that mitochondrial decline is not purely genetic destiny. It's heavily influenced by the absence of what researchers call hormetic stressors, controlled, brief biological challenges that signal to the cell, adapt, upregulate, become more resilient. When those signals are absent, and modern life removes almost all of them, we're temperature-controlled, food-abundant, sedentary for 8 to 10 hours a day, and largely light-deprived in the morning while light-saturated at night. The mitochondria receive a message. Efficiency is no longer required. They downscale accordingly, and you feel it as fatigue that rest doesn't fix. This is the key insight. The fatigue isn't coming from doing too much. It's coming from sending your biology the wrong information. So how do we change the information? The first and arguably most powerful lever is light. Specifically, morning sunlight in the first 30 to 60 minutes after waking. Here's the mechanism. Your suprachiasmatic nucleus, a cluster of roughly 20,000 neurons sitting directly above the optic chiasm in your hypothalamus, is your master circadian clock. It governs the timing of cortisol release, melatonin suppression, core body temperature, and the hormonal cascades that set your alertness architecture for the entire day. That clock is calibrated by one input above all others: the specific wavelength ratio of morning light hitting specialized retinal cells called intrinsically photosensitive retinal ganglion cells. Morning light, low on the horizon, rich in long wavelength red and a balanced ratio of blue, sends a signal that cannot be replicated by indoor lighting at any intensity. A 2021 paper published in Nature Communications confirmed that this early light signal drives a cortisol pulse that peaks 30 to 45 minutes after waking, elevating alertness, focus, and metabolic rate. Afternoon light, by contrast, has the opposite profile and begins suppressing that same cortisol response. The practical implication is exact. Get outside within 30 to 60 minutes of waking, ideally for 10 minutes on a clear day or 20 to 30 minutes on overcast days. No sunglasses. You don't need to stare at the sun. Peripheral light is sufficient. This single behavior has documented downstream effects on sleep quality 14 to 16 hours later, which closes the loop on the next day's recovery. One critical nuance: if you delay this light exposure, say you stay inside until 10 a.m., Your cortisol peak drifts, your melatonin suppression is delayed, and your sleep timing shifts. Even a few consecutive days of missed morning light visibly disrupt the cortisol architecture in controlled studies. Consistency is the whole game here. Now let's talk about movement, but specifically, the timing and type of movement that activates the mitochondrial renewal pathway. There's a molecule most people have never heard of called lactate, which has spent decades being misidentified as a waste product of exercise. It's not. Lactate is a signaling molecule. When you exercise at moderate to high intensity, roughly 65 to 80% of your maximum effort, your working muscles produce lactate, which crosses into the bloodstream and enters the brain. There, it's converted to energy by astrocytes, the glial cells that support neurons, and it activates a gene expression pathway that upregulates BDNF, brain-derived neurotrophic factor, which Dr. John Ratey at Harvard Medical School has called miracle grow for the brain. BDNF promotes neuroplasticity, the growth of new synaptic connections, and the maintenance of dopaminergic pathways that drive motivation and energy. The timing piece matters. Exercise performed within the first 8 hours of your wake cycle, and ideally between hours 2 and 6, aligns with the cortisol and core body temperature rise and maximizes mitochondrial adaptation. Exercise in this window, according to research from the lab of Dr. Sachin Panda at the Salk Institute, also reinforces your circadian signal. If you're someone who currently exercises at 9 or 10 p.m. and then can't sleep, this is physiologically predictable. Late exercise elevates core temperature and norepinephrine at exactly the wrong time. The target is 20 to 40 minutes at moderate to vigorous intensity, in the morning or early afternoon, 4 to 5 days per week. Sleep is where the actual restoration happens, and most people misunderstand what good sleep means at the cellular level. The first 2 cycles of the night, roughly hours 1 through 4, are dominated by slow-wave sleep, during which the pituitary gland releases the majority of its nightly growth hormone pulse. Growth hormone in adults is not primarily about muscle growth. It's about tissue repair, cellular protein synthesis, immune surveillance, and metabolic regulation. A 2020 study in the Journal of Clinical Endocrinology Metabolism showed that even one night of late sleep, shifting sleep from 11 p.m. to 2 a.m., significantly blunted this growth hormone pulse. The window isn't flexible. The pulse is time-locked to early nighttime hours regardless of when you fall asleep. Consistent sleep onset between 10 and 11 p.m. for most adults captures the bulk of that restorative hormone architecture. The protocol is simple, but requires commitment. Dim lights after 9 p.m., keep your room at 65 to 68 degrees Fahrenheit, and anchor your sleep timing within a 30-minute window every night, including weekends. The fourth tool involves the timing of food intake, specifically the practice of a compressed eating window, sometimes called time-restricted eating. When you go 12 to 16 hours without caloric intake, a cellular recycling process called autophagy upregulates dramatically. Autophagy, which won the 2016 Nobel Prize in Physiology. When Yoshinori Osumi's foundational work was recognized, is essentially your body's quality control system for clearing damaged proteins, dysfunctional mitochondrial fragments, and cellular debris. In autophagy's absence, that debris accumulates and directly impairs mitochondrial efficiency. A 14-hour overnight fast, finishing dinner at 7 p.m. and not eating until 9 a.m., is sufficient to activate meaningful autophagy in most people without metabolic stress. This is not starvation, it's a timed biological signal. The final piece, cold exposure, is brief but potent. 2 to 3 minutes of cold water immersion or a cold shower at 55 to 60 degrees Fahrenheit drives a significant norepinephrine release. Research suggests increases of 200 to 300%. That lasts for hours after the cold stimulus ends. Norepinephrine is a dual-function molecule. It sharpens focus, elevates mood, and directly promotes mitochondrial biogenesis, the creation of new mitochondria. The cold itself is uncomfortable. That's the signal. Brief controlled discomfort tells the mitochondria that the environment requires higher output capacity. They adapt. Here is the integration. Morning light within 30 to 60 minutes of waking. No sunglasses. 10 to 30 minutes. Exercise at moderate intensity for 20 to 40 minutes, in the morning if possible, 4 to 5 days per week. Sleep onset between 10 and 11 p.m. Room at 65 to 68 degrees, timing anchored consistently, eating window compressed to 10 hours or less, and 2 to 3 minutes of cold exposure, 3 to 5 mornings per week. None of these are difficult in isolation. What's difficult is that our modern environment defaults to the opposite of all of them. Artificial light at night, no light in the morning, sedentary afternoons, late eating, and thermal comfort at all times. The biology hasn't changed, the environment has. These tools are simply restoring the inputs your mitochondria, your neurons, and your endocrine system have always needed to run properly. The tiredness that feels like aging may not be aging at all. It may be a signal problem, and signal problems have solutions.